start another recording for the 1 second. Oh my god, I need coffee or something. Okay. I will do some push-ups. Alright. Hello, hello, hello. Hello, hello, hello. So here are some notes. Quickly going over what is recombinant DNA, how to make it, how do you identify GM organisms, using putting the gene marker inside them so that they are they depend on one amino acid or chemical or protein, and then we set up two instances where the protein is present or the protein is not present, and then just checking where they're missing present. The vectors in which we transfer recombinant DNA into the target cells, so like as in we take the plasmid from a bacteria and then we turn the plasmid into, we remove it using that, what's it called? Endo restriction endonuclease enzyme. And so then we put in using DNA ligase the desired gene into the plasmid. And then when we have that recombinant DNA, we're going to incorporate that into the host cell DNA by using gene guns, harmless viruses, liposome wrapping, and microinjections. Knockout organisms are to remove a gene or silence a gene rather than instilling a new one. How they're produced, how they're used. Drugs made from microorganisms, drugs from organisms, plants, and eukaryotic organisms. So like drugs obtained from what was it called? Transgenic plants, transgenic animals. How this is made, and then how transgenic plants and animals are actually made. Um, kind of the same thing. Actually, I'm not sure. Yeah, well, no, not really the same thing. Because over here, we're going to be looking at the tomb fascist thing, right? A tomb fascist. So, it is the crown gall, which we extract the cells from and use that to clone and make new plants. And then those plants will be giving you the desired product. Then we have more biotech. So, this is going to be like how to see what genes are going to be expressed more or activated, what genes are going to be silenced when a DNA mutates or gets infected by some sort of mutation. Any sort of mutation, or maybe like cancer cell, right? So like, yeah. Then benefits and risks of GMOs, and that's pretty much it. Describe how microarrays of bioinformatics could be used. Mm, yeah, it's the exact same thing. Man, I should have read the textbook. I feel like an idiot. Because I'm sure this is not the only spot in the text. Like, I read 8C entirely by myself. And I'm sure this was not the only area where they did such a good job in writing. Like I'm sure they did a great job in writing in other places. But when it comes to such theory full or theory backed stuff, I would not really care about reading it from the notes or from this. But maybe I should. I don't know. Well, I mean, it's fine. Anphilic with Frank Sochaki. Sochaki? Sochaki. What a boring cover. Our book isn't like this though. For some reason I deleted the first 2 pages. Actually no I didn't. Sochaki. I'm not really sure how we're supposed to, because we have the exam in 3 hours. So what is our method of doing this? Like, how should we do it? Because we have, like, say, the notes, which are unread, but they're the summary of what we know. There's some parts of the notes which we could read over, like for example, knockout genes I don't remember quite well, and microarrays could use some more work, and how transgenic plants are made. Um, those are some of the few things. Then we have the topicals. No, we know that everything is going to be coming from these topicals. So if we just study right now from the topicals, and I mean that's 99% of the things that's going to be coming in the test. And so first of all, we'll get a feeling for everything that comes in the test. Plus, if there's anything else we don't really know, like then we can just study from here. So I think that's fine. So just to put it again, study from topicals, doubts, go straight to notes. Okay, doubts for doubts. Study from MS. If still unsure, go straight to notes. Okay, I think that's fine. Because I mean, the MS should be enough because I've already studied this topic. Oh, I just cried in your arms tonight. Must have been something you said. I just cried in your arms tonight. Whoa, whoa, whoa. I just cried in your arms tonight. Must have been something you said. Should I walk away? Should I walk away? Okay, so animals and plants can be genetically modified. The recombinant human erythropoietin is a protein drug that can be given to patients with low hemoglobin concentrations. Sheep have been genetically modified to produce RHE in their milk. Explain how sheep could be genetically modified to produce RHE in its milk. Okay, so is this like a is this a so now, where do we originally get the gene from for RHA? From humans? Okay. So I'll originally probably say uh, use endoplasmic sorry, restriction endonuclease. Use restriction endonuclease uh, to isolate uh, isolate desired gene then and to cut an extra. Okay, this is so many steps here. Um, do I just say use a vector? Because it doesn't necessarily have to be that I have to use um, this. There are other ways of doing it. Because we're looking at because like now we have the plasmid, and so when we want to put it into a bacteria, how do we put it in there, right? So to inject it in, we can use gene guns, harmless viruses, lysosome, or microinjections. So let's just say microinjections, right? So okay, I'm not sure. So let's see. Okay, so okay, this is a human one. Okay, this is important. So okay, identify, isolate, cut out. I said isolate the RT gene. Cut DNA sequence using restriction endonuclease enzyme. Okay. Insert the gene into a vector. Insert vector into target sheep cell or tissue. Okay. So if I was thinking microinjections, that would be into the sheep cells. How do I do that? Do I just double click on these? I don't understand. Okay. Again, isolate the RHA gene and cut it out of the DNA strand using restriction endonuclease enzyme. Then insert it into a vector such as like um, see, when we say insert it into a vector, that does not mean we insert into the plasmid. We insert the plasmid into something else. Okay. I feel like some other steps I'm missing here, like um, use the restriction enzyme to cut out a part of the plasmid, and then use DNA ligase to insert the desired gene into the plasmid. Then insert the plasmid gene. Insert the plasmid into. What do you mean the plasmid is the vector? Oh God, is my understanding wrong here? Okay, let us see. Let us see. Let us see. Okay, so if God, so GM sheet. Then we use so okay, I think I get it. So I'm dumb. The plasmid is the vector. So inserting the gene into the vector, which is plasmids, using the DNA ligase, and inserting the vector into the suitable target sheep. So they don't really care how you insert it. Like they don't care how this vector, this plasmid, is inserted into the sheep cells. But it does care that you mentioned that it is to the cell, not just to the sheep. Discuss the risks and benefits of GMO in valuable agricultural plants. Use all the data and knowledge to support your answer. I could see this coming in the test, but you're supposed to just use all the data points that you know. Like um okay, so the risks and the benefits. So this is quite like straightforward question. So um I do wanna try this. So I guess I could make it smaller from here. Let's see. Okay, also we need a new font. This is so dumb. Hmm. Ooh, I like this. New papyrus. Hello guys. Um Dharu is that Bha? Jongri Dhoshor Aghodhao Aghodha. I don't know what that is. Is that Bha? Just little emojis. Oh, okay. Didn't like that. Okay. Zap Dingbats. Oh wait, does it change every time? It does. Okay. Mm, okay. I know it's too dark. Well, you shouldn't be sleeping at 7 a.m. Or if you want to go to the sleeping room. This is the huh? First. I'm spending so much time on this. I'm so dumb, bro. Okay, this is good. Make default. This is so light. Um, Okay, so suppose discuss use all data and knowledge. Okay, well there's no data that we have, so agricultural plants, right? So increased yield, increased shelf time, increased um, resistance to herbicides, herb pesticides, um, increased nutritional value, um, increased resistance to poor growing conditions, abiotic conditions, I should say. Then cons would be risks of genetic modification. So, um, infertility and um, may keep keep slow that may anything else? I mean, that's pretty much it. That all that I can remember. Um, Okay. Shit, I should have talked about. Well, what do these do, right? What does higher nutritional value do? It prevents malnutrition, or it prevents disease, or prevents death. I mean, so you can go that far. It's okay. Anyways, higher yields, less damage by pests, longer shelf life, more predictable food supplies for ever-growing population, improved nutritional content, medication provided through GE foods, genetically engineered foods, reduced cost, reduced use or cost of herbicides and pesticides, withstand environmental conditions. Um, yeah, this is that's the point, right? Like, why is this resistance to herbicides and pesticides used? So that okay, no, it's not. Herbicide pesticide resistance is a thing, but it's also herbicides and pesticides are proteins which are released, like the proteins for herbicide and pesticide like um, resistance or like removal. um, it gets released from the plant itself so that we don't have to use herbicides and pesticides. So yeah, that's actually, yeah, that's a good point. Discussion of risks. So um, increasing the demand, increasing demand agricultural resources and land for agricultural resources and land. How is that a risk? That would be good for, oh, okay, which reduces land available to grow other crops like biofuels. Okay, I see. Increasing monoculture, declining biodiversity, stated effect on local ecosystem, unknown consequences of foreign gene expression. I think this is what I was talking about. Horizontal gene transfer to other organisms and species, aka antibiotic resistance, weed becoming herbicide. Okay, so I need to learn about these risks more because they're not great. I didn't do too well on them. Especially the ones that I did know. This one, we can get rid of this because I didn't actually write that. Um, hmm, I mean, this was probably stated in the question, but I'm not sure if it is. So yeah, 1, 2, 3, 4, 5, 6. I mean, maybe I'll get four marks. But yeah, this is important. So as we increase the like supply of the amount of food that we have, the amount of demand is going. I mean, the amount of demand is going to increase. Like, this is not a sustainable way of making food because we can make more food, sure, but at one point, no matter how much food we make, there is a limited amount of land that we have, right? And so this reduces the land available to grow other crops. Maybe we should just start doing like that thing that some people do where they grow plants on the water beds. You know, what's it called? What is that thing where people grow plants on bodies of water? Hydroponics, aquaponics. Hydroponics. Is hydroponics the future of farming? Hydroponics is a critical component of the future of agriculture, particularly for urban, sustainable, and climate-resilient farming. While unlikely to replace traditional farming for staple crops due to high costs, it offers up to 90% of water savings and higher year-round yields in smaller vertical spaces. R/slash farming. Why is hydroponics not widely used instead of conventional farming? I got some ideas of starting some business around hydroponics, and I'm just wondering why it's not so popular yet. It offers the green theme of the world right now, and the organic food that everyone wants. I'm guessing it has something to do with cost, but from what I got, it does not seem more expensive than conventional farming. So what is it? Thanks for some really good answers. I was expecting someone to tell me it's too expensive and then end of story, but no, I was wrong. Although I got some of those too. Per unit produced, it is orders of magnitude more expensive than conventional farming. That's okay for high value crops like vegetables, but for commodity crops it is not, and will never be a viable option because of infrastructure. I'm not sure what sources you're looking at, but hydroponics is definitely more expensive than conventional farming. Expenses are measured by the square foot, square meter in hydroponics, and by the acre hectare in conventional agriculture. Here are the major areas that conventional farming wins in. I'll list some advantages of hydroponics later. Capital costs. You will pay between $6,000 and $20,000 per acre for farmland in the US, much more than that in a handful of very exclusive locations like Napa Valley, but that's rare. With hydroponics, you have to buy the same land, build some kind of infrastructure on it, install an irrigation system, a fertigation system, etc. Depending on how far you go, this could cost between $0.5 and $25 per square foot, or about $22,000 to $1 million per acre, on top of your land costs. With conventional farming, you may not even have to install an irrigation system depending on where you are located and what crop you're growing. You just have to buy a tractor and some equipment, which could end up being a significant expense for a very small farm, but for a large farm gets spread out and doesn't amount to very much on a per acre basis. Conventional farms are generally highly mechanized, and even the most labor-intensive crops at least have some mechanization that helps with labor efficiency. Labor costs are highly crop dependent, so I'm hesitant to generalize. The most extremely expensive crops that I'm aware of that are grown conventionally have labor costs in the range of $10,000 per acre. But again, that is unusual. The lowest labor expenses might be $75 to $100 per acre for a crop like wheat, if grown on a sufficiently large scale. Even that number really depends on whether you would include the farm manager's salary as part of the labor cost. For hydroponic farming, I had to look up some figures. According to one source, labor expenses for growing lettuce hydroponically were over 19K per acre and 5.7K per acre for hydroponic tomatoes. For comparison, conventional lettuce takes about $900 per acre in labor expense and about $400 per acre to grow tomatoes for canning, ketchup, etc. Fresh tomatoes would be more expensive since they have to be harvested by hand. Even if those figures are off by an order of magnitude, which is I think unlikely, it is still quite a bit cheaper to grow outdoors. I've grown the same crop indoors, not hydroponically exactly, and outdoors on a large scale, and I can say from my experience that outdoor farming was much more profitable. In fact, I'm pretty sure if we had our accounting accurately reflect all the costs of the indoor operation, especially depreciation costs, we were definitely losing money growing plants indoors. We barely turned a profit on it when we didn't factor in depreciation. The outdoor operation more than made up for it and carried the business. Hydroponic growing has one major advantage and major advantage and one advantage that matters in certain circumstances. The major advantage is that the plants are grown indoors. You're able to turn multiple crops in the same space, and you're able to time your harvest to coincide with the highest market prices for that crop. If prices are high and you can get multiple crops at those peak prices, it is possible to make good money growing plants indoors, but you still have to watch your expenses very carefully, especially labor costs, which can get out of control in a hurry and be a good grower. You don't want to make mistakes when you are growing such an inexpensive product in a small space. The smaller advantage of hydroponics is water efficiency. In almost all major agricultural regions, this advantage is almost irrelevant. Water costs in conventional agriculture are between free when irrigated by rainfall and, on the other hand, $1,000 an acre. It's only in certain locations where that isn't the case. Desert environments like those found in Israel, for example. In those circumstances, water efficiency can be an important consideration. Even then, the capital costs could prevent farmers in a huge swath of the world's desert environments from being able to afford hydroponic production. In those circumstances, it is quite typically a bit cheaper to import food than trying to grow it locally. Yeah. Man, this guy writes so much. I can't read this much. It is surely not more green than organic farming in the ground as plants naturally grow. There's a huge lack of terps. You just get big plants. Huge lack of because there's a huge lack of interpreters. I work at Amazon. I know what y'all order. The home hydroponics kits Aero Garden has always been selling much more than any other hydroponic stuff, and I will say it's close to regular gardening. I suspect that the reality is gardening overall is hard. Small scale hydroponics may be sold as gifts, but not enough that people are picking up the hobby. Paired with paired with the fact that nobody has the time to grow and cook their own food. Also, unlike something like 3D printing, it's boring. Thankfully, I have a crew of nerds who love to help me on these endeavors, so my solution is to just buy a bunch of these gifts and try to build a local community. Everyone has to start somewhere, so making it an easy start is to get into it is the best method. Buying extras and gifting them my community, it's slow, but it's catching on. Even on a small scale, I had to pull back from hydroponics. We rent, so we can't install a decent water filter. Getting in distilled water was costing way too much. The nutrients were expensive to get in Canada where I live, and we barely had any selection. Trying to figure out why I'd get blossom drop or nutrient deficiencies, despite adding in the nutrients correctly, was frustrating. Taking apart and disinfecting the system between harvests or when there was an issue was a ton of work. Now I have buckets of good soil, giving them tap water and initial fertilizer I can get my hands on. The only thing that grew better in hydroponics than in the soil was lettuce. Those I did in big jars, covered in till fall and the crafty method, and it was kind of amazing. Well, Ooh, there's quite a bit of shade structure. I've been thinking about putting a shade structure over there. Oh my god, bro. It's a pipe dream, snake oil, and artificial faith for those who know little but have big faith in technology. So my comment was careless and lazy. Hydroponics is a proven method for growing plants growing in a so no shade thrown there. However, vertical farming is inherently scalable due to major costs coming from electricity requirements. I only meant to shit on the idea of scaled vertical farming. I appreciate your time. Anti-vaxxer base. Suggestions to keep mulch in place without a retaining wall. Trench. Landscaping. Oh no. That's so funny, bro. What the hell? Planted 56 cypresses. God, this looks so dumb. All my friends and family are telling me I made a horrible mistake. I like to disagree with them, but inside I feel I logically did make a mistake. Right now I'm deciding with my family that the best decision is to remove them before I regret them years later. And they say this doesn't make sense in the amount of space in the type of house. Some of them already have brown leaves at the bottom part where you see cypresses under Anyways, do you think I should remove them or that I am Picasso and it's beautiful? I'm sorry, but this would be absolutely hilarious if you keep these alive to the point of having a literal wall of cypress trees. It's like an impenetrable fortress or something. Maybe that was the point of planting them. I'm not seeing why OP planted them or why relatives are saying it's a mistake. There's still room to add more. Your property's future. Let's see. Oh my god. What the hell, man? This looks hilarious. Oh my god. Oh my god, from the side, it's so funny. Can it go here? Yes, you can see it from the side. Amazing. That is beautiful. Holy hell, I didn't realize how ridiculous the end result would look, but that's some Dr. Seuss shit right there. Your ski phone made me laugh so hard, man. It was really Dr. Seuss. Initially, I didn't understand the problem with open spoils because I know nothing about trees, but god, that's hilarious. Hot. Did you Google your mature size? Do that before choosing a replacement. What a great post. Thanks for the shout out. I didn't. I looked it up. Nothing surrounding your home in a dome of darkness. The AC savings gonna be wild. All savings can go towards therapy for year-round seasonal depression. Obi needs to pre-game vitamin D. It's fun. First you're away in your own Cyprus city-state called Cyprus. I thought that this was a ship post. It was until picture 5-7. That's when I realized that those were in fact not on the roof. Wait, really? Okay. These ones have already started drying. And that's a huge lot. I don't got that. I'm freaking dying thinking about you having like a two spots surrounded with trees as well. Your neighbors having to explain every guest that comes over that you're at the home. Keep them just to be neighborhood lore. Reddit lore. This guy sounds like me. Lamar will be hilarious. Remind me 10 years. Check this guy's impenetrable wall of cypresses on 3 acre footprint. Remind me in 100,000 years. Imagine looking out your window at home and always see the wall of green, and that's looking out from every window. Literal dream home. It's fine if you got huge ass property. This is going to look like jail, but green. Is that a problem? In a perfect world. That's amazing. Yeah, they need a mod. Yeah. Realizing that 2041 is only 15 years away makes me nauseous for some reason. My unborn child will be driving in the 2040s. Will still be circling down the trains. Oh, God. Stop. First mistake, planting 56 cypress. Second mistake, putting down landscape fabric. Third mistake, using artificial turf. Sincerely, a professional horticulturist. That house is going to have a very strong character in a few years. I love it. One with the whole family. Now my grandfather had this cool hedge for like 50 plus years. They grow slow and every few years. Don't let it grow over 6 feet. You are assuming that you will do nothing. You have to touch the sky. 6 feet isn't easy and quite to maintain. Maximum above 7 feet. You want to with cypresses, how much it's all cost. Oh god, man, this is so funny. Ah, oh, wow. Saw this house with a massive cypress, trying to imagine what it would look like with 55 more. Oh, oh. Oh my god. Oh my god. Oh. That person will never live that down. Oh my god. Planting a cypress in my yard every day until Reddit says they're perfect. This is a callback to the other post where planted a fortress around his house. I really hope my old ass is around to see his progress reports. Man rescued me a helicopter to being trapped inside. It's the only reason I'll be staying on Reddit until I die. Since nobody linked to the post is referencing that. Hmm. 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 It was no accident. Just keep them off the route. I think that other guy's pulling them out because you guys bullied him. Come on now, we didn't bully him, we unschooled him. That's what it is for. Not because we bullied him. First of all, it was some good natured ribbing, just like you do when one of your friends does something stupid. Second, the dude planted 56 cypress trees around his property, pulling some out is the correct move, not to be completely left out. Oh wow, that looks really good. Yeah, it's not bad actually. I think I will think about that post for the rest of my life. At least it's got like so many upvotes, you know. Breaking logic, breaking one before it, then you can just line up your shiny stripe. Check the shiny stripe. I was actually flying in for my body. I found I guess far from the service from huh? Your art is obviously shade, which tremendously helps its strength. Watermelon looking at the art. Yeah, it's a bit excessive now. Five species were extinct for this specific mound. I know, right? Story straight action, but yeah, if you wanna look at how your brother straight your pick, I know Cinderella. I wish she did best. Fourth light stripe from the off. Oh, really? Can you teach me how to perfectly match my stripes? And how do you know I even know on the side of fence? You don't, but you're not, because I don't. But hey, come on, I can give you the bracelets on no one does. Call him till you come out. I don't know the stripes on that for the day. You was my story to find fancy on the phone. Everything you can. That's how I talk to. Lighten up, Francis. Figures of these men like that, you're fragile. He was obviously joking. Yikes. Okay. Bro. 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 Oh god, bro. I need to show Dutch this. Oh my god, Dutch would die. Oh, he would die. Oh god. I was imagining what it would look like with 55 more. Abruptly cut for trees. Really accentuates the holes in the gas meter. Oh my god. Poetry. Oh, I need to study, bro. This is so funny, dude. Ah, this is so funny. How do we get there? We went from reducing land available to grow from crops, so then I wanted to look at hydro farming. And then from hydro farming, I went from hydro farming to one of the people who was farming. No, he wasn't farming. He just hated on it, and then he's on the landscaper subreddit, and then I went to the landscaper subreddit and then I found this. Yeah. Oh god, my heart. I can't handle this. Oh. 8 o'clock. Time flies. Ah, okay. So yeah, mm, that should hold the point pretty well. That as the demand grows, the amount of land that will be able to supply, that will be able to be used for agricultural crops, is going to decrease, is going to fall. So that's a concern, that's a risk. The second risk is monoculture. So if you keep on growing plants which are all the same, the biodiversity will decline. As the biodiversity declines, then um, although like we are growing like essentially super plants which are going to survive in drought conditions, etc., etc., etc. There is a thing to be said that if you have a monoculture, then that is going to it's like if you had an education system which only favors one type of individual, so any other types of individuals would not survive there, or would not be able to excel there. And so as a result, any other individuals which stem from those individuals which aren't able to have success will also not have success, right? And so in the long run, that has a pretty big effect on the ecosystem. Now, when it comes to agriculture, I mean, I don't think ecosystem really matters. They probably would want to control environment more than anything. But yeah. Unknown consequences of foreign gene expression, long-term effect unknown. So yeah, the long-term effect of doing these gene expressions is unknown. Apparently, this is the first time I'm hearing about it, right? So in the long run, does doing gene expression on a plant or animal um, affect it? Maybe another discussion of the risk is like stress or like animal testing, like plant testing. Maybe you could be harming plants and stuff like that, but I'm not sure. Next is gonna be horizontal gene transfer to other organisms and species. So this is important. The term that they use, horizontal gene transfer to other organisms and species. This is what I meant. Um, Marker genes coding for herbicide resistance may spread into the environment and be inherited by wild plants. So the gene may transfer to other organisms and species. No, Dutch isn't in the server anymore. Ah, is he? Oh, yeah, he is. Oh, dude, that guy was so handsome. Like, really, really handsome. Last time we spoke was April 17th. Not too long ago, but yeah, quite a while ago. He has 47K messages here, so a lot. Something or something. Maybe I was a youth for my fault, but I've been really clear. On the topic of African cheers. Actually, now we can just feel about the grass, the shovel, on the open heart surgeon. Also, come back. On the map, School that this will be all Bulgar ancestry and trying to without them. Are you asking her dirty girl full that is that you? Oh, someone just like that, so you get the look. Oh damn, that is pretty good. I like to get down, I like to boogie. Stay away from my wife. Damn, next time you better so delete that. Disco shit. The necessity of following the curve of the road and matching it going over just the ball. Yes, sir. One way 1 week, the other way the next. That's how I'd more if I was listening still tip on Mike Jones. This is sick, I feel. Like the nightlife. I love the boogie. This guy high tensions. Moyana as the animals. Don't knock it till you try it. Show off. Describe as Jason Bird. This is where I want to discuss. Honestly, this picture is from 2 3 years back. Before I was really the problem it is now. While I would pay heavy like for a cash course on how to lay these lines and then I've tried it's gonna get right. It's pretty basically like I got one curve 1 week, the rest of the next. The big thing for it is to like right. Get the curves seem like already this one. We know like weeks backyard using this technique. We just follow the curves by from the concept school that fixed. It says you just follow the predetermined school path of the road. Yeah, it's really well. I love this guy, he's so chill. Or maybe we can throw him. Who's Mike Leake? Mike Leake. Leach? Leak. American college football coach who primarily coached at NCAA. Nicknamed the Pirate. He is the two-time National Coach of the Year, three-time Conference Coach of the Year, and mastermind behind the NCAA record-setting air raid defense. Let's see. He has books on hematology. Okay. I need to study. Let's go tonight. Let's go tonight. One potential treatment for AAT deficiencies using adenoviruses uses adenoviruses to genetically modify cells in an affected person. This is probably the vector though. Contains double-stranded DNA. It uses to treat AAT deficiency in a person. Double-stranded DNA. Does that mean that it contains exons? But humans could probably remove those exons, so. Eight. Oh, yeah. Okay. So I can't tell. Are we just how adenoviruses can be used to treat AAT deficiency? So clearly, adenoviruses contain genes in their double-stranded DNA which codes for protein which has AAD in it. So how are adenoviruses used? So I'm guessing are adenoviruses harmless? Wait, use adenoviruses to genetically modify cells in an infected person. So okay, so we would use DNA helicase and then DNA helicase and then restriction endonuclease to remove the gene which codes for AAT in adenoviruses, then insert it into a vector and then insert the vector into the human through microinjections, into a human cell through microinjections. Is that it? Identification of corrected AAT gene, description of one enzyme to insert gene. Yep. Adenoviruses can enter host cell, combine. Okay, wait. Adenovirus acts as a vector. Insert the gene into the adenovirus DNA using DNA ligase. Oh, okay. Wait. cut the adenovirus, sorry, cut the adenovirus DNA using the using the restriction of the nuclease. Insert the gene adenovirus DNA using DNA ligase. Use the adenovirus as the vector. Adenovirus can enter host cell and combine their own genetic material with that of the cell. Therefore, a functional AAT protein can be produced. So this is the important part right here, where adenoviruses can be acting as the vector. They can combine their own genetic material with the cell's genetic material, like the proviral DNA, and so a functional AAT protein can be produced. This is important, that this ending point, that a functional AAT protein can be produced. Vitamin A is an essential part of the human diet. What is anti or nuts? Rice is a staple food in parts of the world where a lot of people have A deficiency. Rice contains no B-carotene or vitamin A. Cells in the human intestine can convert B-carotene from the diet to vitamin A. Okay. Some soil bacteria can produce large quantities of B-carotene. Explain how genetic engineering can produce rice that contains B-carotene. So again, isolate the gene in soil bacteria that produces B-carotene, then that produces the protein for B-carotene, then use restriction endonuclease enzyme to remove that gene, insert the gene into a vector, insert the vector um, into humans. Yeah. Making beta carotene bacteria. Cut DNA sequence, extract beta carotene using restriction enzyme. Okay, so this is important. We're saying cut DNA sequence. Okay, it's always cut DNA sequence. Cut DNA sequence, cut DNA sequence, cut DNA sequence using restriction and the nuclease enzyme. Insert gene into vector. Gene gun or microinjection. Grow. Okay, this is the last point I didn't write. Grow rice and select from modified rice plants with high yields of beta carotene. So this is how we like identify the plants which are GMOs and selectively grow those. That's basically what they're saying. The possible risks of growing genetically modified rice plants. Okay, so it's the same ones we wrote here. um, okay, and you have to remember these as well. Okay, cool. So what was it? It was the ones in the 6 marker that it talked about how demand will increase for genetically modified plants, but the amount of land that's present, uh, the amount of farmland that is used to grow plants is limited. Okay, then the amount of then uh, this promotes monoculture, which would reduce the biodiversity of the ecosystem, or which would affect the ecosystem negatively. I don't know how though. I'll have to look at that. Then last one was um, horizontal gene transfer to the environment or to the ecosystem, such as antibiotic resistance and pesticide herbicide resistance. Um, and also the other one, which was long-term side effects of the um, the genetic engineering is unknown. Um, so okay, but in the terms of genetically modified rice plants, right? So long-term effects of consuming it is unknown. I mean, that's not true though, but okay. Accumulation of genetically engineered material in the thing, crossbreeding with other things, pollen transfer, altered gene is found in other species. Unknown long-term side effects of eating GMOs. Oh, there you go. Allow. Okay, this is what? Where did this come from? allows production of superpests. Oh, okay, yeah. So we were right. So pests which are resistant to herbicides, right? So these are superpests. So the idea of superpests or superweeds is important, yeah. Um, Gene transfer should be okay, but this is what it's caused by. Crossbreeding is caused. I mean, gene transfer is caused by crossbreeding. Accumulation of genetically engineered material in the environment. Um, okay. So just how one of these risks can be reduced. Um, uh, Growing them in like greenhouses away from like the outdoors. Grow in secure greenhouses or grow separately GM and non-GM crops. Okay, this is important because this was there in the note. Describe how microarrays can be and bioinformatics can be used to investigate the genetic basis of Parkinson's disease. So I guess we take a sample of healthy cells, or healthy somatic cells, and a sample of somatic cells infected by Parkinson's disease, and we use a microarray to identify the genes which are expressed more and which are silenced due to the gene, due to the disease, and yeah, that gives us more information. So microarrays allow identification of actin genes and gene transcription. The activity of okay, so I basically said that, not, but I didn't define it, so we should define it first. The activity of many genes can be analyzed in a single sample. Um, the activity of many genes can be analyzed in a single sample. Okay, that is true, yeah. So advantage, okay, is this the advantages? No, they're just saying it because like LAMIO, okay. Let's go tonight. So this is just one of the advantages, I guess. By collecting information about genetic differences from many individuals, bioinformatics computers use to analyze the data. Um, Differences between healthy and okay, yeah, there you go. Differences between healthy and individuals can be identified. Um, Analyzing the data because there's going to be, yeah, right, I remember this. So we use bioinformatics or computers to analyze the like the orders of magnitude of data that is formed from research from this. So this is pretty big. Collecting information about genetic differences from many individuals with or without. So genetic differences, information about genetic differences. What is the difference between these 2 points? Um, Many activity of many genes can be analyzed in a single sample. Microarrays can allow the identification of gene transcription, how the gene transcription changes. Okay, I remember there was one thing I didn't know. I said something like this, and then something I forgot. I don't know. Was it the thing up here? Um, I don't know. Anyways, mutations in other genes can result in blindness. Describe how these genes can be identified. So we take okay. So using a microarray in bioinformatics, we can take a group of healthy cells and group of diseased cells or mutated cells, and using a microarray, we can identify the genetic differences between both. Um, uh, uh, we can identify the transcription or the activity of many genes in healthy and mutated cells. And okay, now we can look at, okay, do we have to describe so if like genes which are red are going to be genes that are involved in it or something. It's only 3 marks though, so I don't know. Using microarray techniques to identify genes that are actually transcribed in the eye. Sequencing the genome. DNA sequencing, DNA profiling. Yeah. DNA sequencing the genome of individuals with inherited forms of blindness. What's called molecular. Compare the sequences for active genes from a large number of individuals with and without blindness to identify mutations. Yeah. I don't know if I would get that many marks on this question. I'm not very good at them. Describe the roles of the enzymes involved in stage 1. They didn't really show us, but it will again. Describe the structure of the modified plasmid used in stage 2. Definition of recombinant DNA. A small circle of DNA or plasmid containing bacterial survival genes and protein animal genes, marker genes as well. For the last 2 questions. Oh, okay, it's right here. So, some organisms have been genetically modified to produce proteins including hormones and vaccines. And man, why is this chapter my favorite chapter? I mean, technically, it is kind of like everything we learned before were kind of like the prerequisites you need to know for learning this stuff, right? Like, it would be hard to know this stuff without knowing the early prerequisite stuff. So, yeah. It's like being able to see the applications of everything that you studied. Like, hey, I understand why this happens and this happens and how this happens and this happens, you know? So that's probably why. Some organisms have been genetically modified to produce protein. Okay, flow diagram, doubtful. Stage 1, gene to produce a protein removed from an animal cell and inserted into a plasmid. Okay, gene to produce a protein which is removed from the animal cell and inserted into a plasmid. So describe and explain the roles of enzymes involved in stage 1. So yeah, so restriction enzyme is used to isolate or cut the DNA and extract the gene which produces the protein from the animal DNA. Restriction enzyme also used to cut the plasmid of the cut the cut the DNA of the plasmid. Then DNA ligase enzyme is used to fuse or um, insert the desired gene into the plasmid. So reference to restriction enzyme to cut a gene out of animal DNA, IDF amplification of using DNA polymerase and PCR. Okay. Enzymes open plasmids, so they don't refer to DNA restriction enzyme here. Oh no, okay. And to also open the plasmid. Okay. Then same endonuclease to produce sticky ends. Okay. Hydrogen bonds between bases of sticky ends. Ligase to join gene to plasmid, reference to phosphodiester bonds. So 5 marks. Is all of them giving you 1 mark? Because then I would get 1, 2, 3, 4, 5. But these are pretty important as well. So if you want to have more of the specific gene, we would use PCR technique to do that. Then we also know that endonucleases produce sticky ends at the sticky ends at the sides of which the DNA was cut. And hydrogen bonds form between the bases at sticky ends. And when we use the ligase to join the gene to the plasmid, the desired gene to the plasmid, we form phosphodiester bonds because remember, these are all bases at the end of the day, right? So phosphodiester bonds are going to be the thing. Yeah, not peptide bonds, phosphodiester bonds, right? Yeah. Then describe the structure of the modified plasmid used in stage 2. Modified plasmid incorporated into bacterium, which is then introduced into the plant. Modified plasmid incorporated into bacterium, which is then introduced into the plant. Um, Yeah. So describe the structure of modified plasmid. Um, it is just like a circular piece of DNA with some alien DNA in it. Um, Small circle of DNA or loop containing okay, and the marker gene, right? Marker gene is also inserted, and it contains bacterial survival genes and okay, bacterial genes and protein animal genes, right? So the animal gene, that's going to be the alien gene, and the bacterial genes. It's going to be primarily the bacterial gene as well as the marker gene. Okay. So this is important. This is actually quite important. That's it. Looking good. Looking good. Honestly, not too bad. Not too shabby. Not too shabby. Oh. I'll be back. Not too shabby. Totally did not spend like just 30 minutes. Was it? It was a more. I think it was more. Just like scrolling on Discord. Like, oh my god, it's so fun going through like old fun conversations. I can't be the only person who does this, right? It's so fun. Mm, it's so much fun. Maybe one day I'll be able to go through those chats again. I mean, I don't even know how to. That's the thing. Even if I wanted to listen to all like the VMs and stuff, like if I were to type like just like it doesn't even pop up. So yeah. Um. Yeah. It's okay. Like it's fine. It's fine. I'm not really that bothered about it. Not as much as it was before. She's quite like I think she's a pretty like imbued. Imbued is imbued the right word? There's like a comedian and definitely is pretty instilled into the community. I would say. Way more than me. Way more than me. But I mean, that's not a very high bar. Like Ariane is way more than me. Um, Probably the guys like this like are like the most. Um, and then like Ira, then but like people like Suri, I don't know. Whoa, Lance is on Instagram. They're all private. That's so cringe. It's fine. But yeah, they all have like they're pretty like actually I don't know, are they? Like is there like it's easy. Yeah. But Lance not really. Lance would be more of his in his friend group. Get your way up, cause it's time to go beast mode. তোমার বেশি ওর তো ক্লাস আর তোমার তো কেন প্লিজ স্টপ কেন আর তুই বস ওই আসবে যা ফাতিমা আই এম গিভিং ইউ 5 মিনিট ফাতিমা এরপর আমি পানি নিয়ে ফাতিমা যা লেটে থাক খাওয়ার জন্য Oh my god. Oops, I left the microphone on, didn't I? Sorry. I had lunch. I'll be back in a bit.